Okay, so let's set the scene. Your cell just went through all the trouble of glycolysis, right? It split a glucose molecule and now has two molecules of pyruvate. And over on the other side of the room, you've got the Krebs cycle, all warmed up and ready to generate some serious energy. But the pyruvate can't just waltz right in. There's a problem, a kind of compatibility issue. So, yeah, what's the holdup? The holdup is that we need a bridge. We need a very specific enzyme-controlled molecular bridge to get from the end of glycolysis to the start of the Krebs cycle. And that bridge, you guessed it, is called the link reaction. And here it is, the official exam-ready definition. The link reaction is the process that takes that pyruvate and, with the help of enzymes, completely transforms it into a different molecule called acetyl coenzyme A. Its entire job is just to connect these two massive stages of respiration. You can think of it as the ultimate metabolic middleman. So let's break this down into three key things you absolutely have to know. One, it's enzyme-controlled, which is pretty standard for reactions in your body. Two, it's the link we've been talking about. And three, and this one's a really big deal, it only, and I mean only, happens when oxygen is around. It is a strictly aerobic process. We'll get into why a little later. So, where does this all-important conversion take place? Well, the location is just as critical as the reaction itself. Let's pinpoint exactly where this bridge gets built. It all goes down right here, inside the mitochondrial matrix. That's the goopy, fluid-filled center of the mitochondrion. So we're not in the cytoplasm anymore, where glycolysis happened. We are deep inside the cell's power plant. Getting that location right is absolutely key. And that location means our pyruvate has to go on a little journey. See, it was made out in the cytoplasm. So to get into the matrix, it has to be actively transported across the mitochondrial membranes. The cell actually has to spend a bit of energy just to pull it into the right room so the link reaction can get started. All right, our pyruvate has arrived at the party. Now it needs a total chemical makeover before it can get its ticket into the Krebs cycle. It's a three-step transformation, and lucky for us, there's a fantastic little trick to remember it. Just think of the letters DOA. The whole process breaks down beautifully into these three steps, which you can remember with DOA. That's D for decarboxylation, O for oxidation, and A for acetyl-CoA formation. Let's quickly walk through what each of those means. First up, D for decarboxylation. It sounds complicated, but it just means removing a carbon atom. Our 3-carbon pyruvate gets one of its carbons snipped right off, and that carbon leaves as a molecule of carbon dioxide. And yep, this is actually part of the CO2 you're breathing out at this very moment. And just like that, our molecule is down from 3 carbons to 2. Next up is O for oxidation. And here it happens through a process called dehydrogenation. Again, it's exactly what it sounds like. Hydrogen gets stripped away from our 2-carbon molecule. But it doesn't just float off. It gets picked up by a special coenzyme called NAD, turning it into reduced NAD. You can think of reduced NAD as a tiny charged-up battery, just holding on to that energy to be cashed in for a big payoff later. And finally, we have A. That leftover 2-carbon acetyl group is pretty reactive. So to stabilize it, the cell attaches another molecule called coenzyme A. And voila, we have our final product, acetyl coenzyme A. This is the molecule that has the all-access pass, the official golden ticket into the Krebs cycle. Okay, the makeover is complete. So what's the final loot from this chemical transformation? Let's do a quick tally of everything we've made. It's a really simple and clean ratio. For every single molecule of pyruvate that goes into the link reaction, we get 1 molecule of acetyl-CoA that's ready for the Krebs cycle. 1 molecule of CO2, which is just waste, and 1 of those little reduced NAD energy carriers. Simple as that. But wait a second. You always have to remember where this whole thing started. Glycolysis splits 1 glucose into 2 pyruvate molecules. That means for every single glucose that you start with, the link reaction has to run twice. So you have to double everything. That means 2 acetyl-CoA, 2 CO2, and 2 reduced NAD for every 1 glucose. Forgetting to double that is such a classic exam mistake, so don't let it catch you out. And now for maybe the most important number in the link reaction. 0. Zilch, nada. That is how much ATP is produced directly in this stage. Absolutely none. This is purely a prep step. Its whole job is to get the Krebs cycle ready and to send those reduced NAD batteries on to the final stage. There is no immediate ATP payoff right here.
So if it doesn't even make any ATP, why are we dedicating so much time to it? Why does this little bridge matter so much? Let's get right to the bottom line.
This slide right here is pure exam gold. Why is it essential? Well, first, it makes acetyl-CoA, which is the mandatory fuel for the Krebs cycle. The cycle literally cannot start without it. Second, it makes that reduced NAD, which is carrying high-energy electrons to the final stage for that huge ATP payday. The bottom line is simple. Without the link reaction, the main energy-producing stages just grind to a halt. It is completely non-negotiable. Okay, let's solve one last mystery. I said at the very beginning that this process is strictly aerobic. But if you think back to the steps, we never actually used an oxygen molecule, did we? So what's going on? Why does it need oxygen to be hanging around in the cell? And this really shows you how all the stages of respiration are connected. See, the link reaction needs a constant supply of regular NAD to pick up those hydrogens. The job of recycling the used reduced NAD back into fresh NAD, well, that job belongs to oxygen, way down the line in the final stage. So if there's no oxygen, that recycling process stops. The cell runs out of fresh NAD, and the whole link reaction just grinds to a halt.
So if you ever needed to wrap this entire process up in one perfect, information-packed sentence, this is exactly how you do it. Let's just look at the first half for a second. And here is the killer ending, which supplies hydrogen to oxidative phosphorylation. That last part is so important. It shows you really get it because you're connecting this little intermediate step to the ultimate goal of respiration, that final stage where all those reduced NAD batteries finally get cashed in to make a ton of ATP. Let's put it all together one more time. In the link reaction, pyruvate is decarboxylated and dehydrogenated in the mitochondrial matrix to form acetyl coenzyme A, producing carbon dioxide and reduced NAD, which supplies hydrogen to oxidative phosphorylation. Seriously, memorize that sentence. It hits every keyword, it shows you understand the whole process, and it explains its purpose. It's the perfect summary of cellular respiration's essential, unskippable checkpoint, the tiny bridge that really does make all the difference.